What is going on? It is March 4th, 2022, 11:05 a.m. and I am here again today, as I will be every day. Before I continue with talking about the pathway to discovering eternal life, which many have gone down the same path, I'm just traversing it now and hope to perhaps one day traverse a little farther than the last explorers or Ponce de Leon or whoever. So hit the like, subscribe, and all that. And it's important to keep me. I'm motivated anyway, but it's important to give my channel some visibility and also help me help move this forward. And it's going to be a long time, as you know, but or as you can guess, but I think the reward will be worth it at the end. I am today going to talk about glucose transportation. Okay, it seems that I've veered off topic a slight bit from yesterday's topic, so I think I should just abandon the idea at this point of cohesively like going from topic to topic, from my original goal, and I'm gonna just talk about metabolism actually quite in depth because this is, if I want to get to the DNA, the genetic level, right? But it's really important to get, I think, cover everything regarding metabolism, because metabolism is the primary process by which a cell survives and lives. So in order to really understand the processes that can get affected by issues with DNA in terms of cellular lifespan and whatnot, we should really understand what's going on in the metabolic pathways. And the good thing is that people have covered metabolism quite in depth, because people are exercise freaks, and also because metabolism, glycolysis being the first step, which is admittedly a bit of a daunting step. So I've decided to break down my study of glycolysis a little differently than just kind of throwing all the intermediate steps of glycolysis at you. But it is a bit of an intimidating step, but understanding all these steps bit by bit will help us really create the picture of life by the end of this session, and like what sustains it. And so I've decided to dub March as Metabolism March, because that's going to be, I think, my entire area of focus for all of March. I think January going into February was actually a lot neural stuff, or at least touching on neurochemistry. And so this month I'm going to focus on the biochemistry of metabolism. And to be honest, like, even besides the understanding the precepts to the life extension, by itself, metabolism is interesting because the metabolic glycolysis is present in even the most archaic creatures, right? And it's kind of evidence, like the process is mostly similar. It's evidence in a way that, of evolution, right? If we were originally far back billions of years ago descended from prokaryotic organisms that use glycolysis, very basic process to generate the energy they needed to take on their life activities, then we still using it today, is a testament to how we are connected to that original construct. And obviously we use more complicated mechanisms now, like the Krebs cycle and whatnot, in our own bodies and our own cells to generate sufficient ATP, because we require significantly more ATP than a prokaryotic organism, right? We still will use glycolysis, because the products of glycolysis, besides ATP, are needed to power the Krebs cycle itself. So yeah, anyway, that's my precursor on glycolysis and why I plan to really delve down the entire metabolic route. But the way that I'm going to do it is talk about it from the very first step down to the intermediate steps within, and all the way into the cell and everything that the cell does, etc. So the first thing to talk about is the glucose transportation, because metabolism starts with glucose, sugar metabolism, right? But metabolism involving glucose is a primarily important process to energy generation, right? And glucose is the molecule which glucose or sugar is the molecule which powers glycolysis, right? And glycolysis is the breakdown of glucose and the conversion into pyruvate. Okay, if you remember that from school, that's kind of what the whole that's the end product of the process, plus 2 ATPs and some other molecules which we'll get into. But let's start. If we need glucose to start the reaction in a cell, right? The first step is how do we get the glucose into the cell, right? I'm not going to go over the glucose molecular structure right now. I will go over it later, but because I want to jump into the actual process. But glucose can be represented by these. It's a hexameric sugar, right? And it's glucose, right? So it's like basically a ring sugar, 6-ring sugar. And this is the oxygen, right? And you have the glucose molecule. Essentially, you need it to get inside the cell, right? So what is the situation going on? Why can't glucose just go inside the cell? So one thing to note, glucose is expected to be in abundance outside of a cell, and that's the whole basis of this transportation mechanism that I'm going to talk about, because in general, there's going to be a lot that the cell is going to assume that it can take in at any time, because it's going to constantly need to power itself, right? And glucose is blocked from going into the cell normally by the phospholipid bilayer here, right? And glucose can't go because glucose is a polar molecule, and you have the phospholipids being negatively charged, so they're polar, so they would repel the glucose. That's one reason that glucose can't just pass through the phospholipid bilayer, because a lot of diffusion or passage into a cell is done through passive transport, right? Or even penocytosis, the cell like drinking or gulping little areas around it, or again molecules that are small enough and non-polar enough to pass through the cell will be able to go through the phospholipid bilayer, but that doesn't actually include a ton of molecules, right? Glucose is one of those that they aren't charged in the right way to pass through the cell, plus they're also too large. Glucose trying to get through, even if the glucose was charged properly, the glucose trying to get through these phosphates would encounter what's known as steric hindrance, and that means the glucose chemical structure would literally physically be blocked by the phosphate structure. So that's interesting, because it's like normally you think of chemical reactions being electromagnetically involved down to the atomic level, but this is an actual physical barrier, right? And and these are on nanoscale things that are also blocked by physical barriers. So that's very interesting, isn't it? Anyway, so the glucose can't just go through the phospholipid bilayer for those two reasons. So the glucose must get into the cell through a mechanism called GLUT, or glucose transporter. Glucose transporter, there are several kinds in your system, and these are glucose transporters are transmembrane proteins that live embedded within the phospholipids bilayer, and they have a cytosolic part and they have a extracellular part, and essentially what happens is that when glucose is nearby, these transporters are normally opened in this T1 configuration. And by the way, I'm only talking about narrowing my focus here. I'm only talking about the basic GLUT transporter. There's multiple types which live on other organs, and there's ones that are not as simple, as in they depend on insulin. And the second thing is this GLUT transportation. I'm only talking about at this point entry into a cell. I will talk about exiting a cell later, and that's when the cell is actually producing glucose, or has to allow it to leave the cell, and that transportation mechanism has other factors. But just the basic entry of glucose in the cell essentially is going to be facilitated by interaction with this GLUT, which has this open kind of pliers conformation and has a section in the center of the GLUT mechanism to support the glucose fitting in. Once the glucose enters through random diffusion processes, this GLUT transporter will have a conformation change. So that means, like normally proteins, when they get activated, they have some sort of conformational change which cause them to behave differently and finish the rest of their function. So that's what happens. The glucose goes into the GLUT transporter. The GLUT transporter then turns into an upside down plier type structure. Whoops. And then that glucose is allowed to leave into the cytoplasm, where then the next step after the glucose is in the cytoplasm is the glucose phosphorylation into glucose 6-phosphate through the interaction with hexokinases. Anyway, that's we'll talk about that later. But yeah, so that's basically the mechanism. And if we want to look at like the GLUT transporter molecular structure, I think it's interesting because here comes the glucose molecule. There are a lot of different, I'm going to go into the molecular structure of GLUT later, but you can see that there's a lot of like alpha helix chains and there's some beta chains as well. We can go into the specifics later, but in fact I do want to go into specifics later, but we're going to look at the crystal structure to kind of like understand more about how GLUT functions on the chemical and the amino acid interaction level, on the peptide level, because that's really cool. So that was a quick thing on GLUT, and just to give you a little bit of an indication where these glucose transporters live. There's other things to note. These GLUT transporters, the basic one that I'm talking about is falls into the category of passive diffusion or facilitated passive diffusion. Okay, so basically it's facilitated means that these GLUTs need to exist in order for the glucose to go into the cell, but the GLUT transporters themselves aren't activated through ATP, so that means they don't have energy requirement. They just happen because glucose happens to accumulate outside of a cell and it can go inside a cell. Now that's a little different when glucose is exiting a cell, because there's a co-transportation mechanism required, because the glucose concentration is larger outside the cell, but the cell still has to excrete glucose, so it's no longer just passive facilitated diffusion. There has to be active element to that, but we can talk about that later. Again, this GLUT transporter is passive diffusion requires no energy. Facilitated passive diffusion, as I like to call it, requires just a channel which interacts, but still no energy. And then there's active transport, which requires energy. Okay, and just